Hello, this is the third and last part of the molecular basis of inheritance. And we're going to start talking about complementary DNA or cDNA. Actually, complementary DNA is DNA that is formed on top of a molecule of messenger RNA. You remember when we talked about the retroviruses, we said that retroviruses have a genome that is made of RNA. So actually, it is a genome that is made of RNA. And those retroviruses have a problem, a major problem, that they want to insert their genome into a host that has a DNA genome, and therefore they have to convert their RNA into DNA, and that's why they use the enzyme that is called reverse transcriptase enzyme that does the reverse of the process of transcription and that builds a complementary molecule of DNA on top of the molecule of RNA, and it is built in the direction 5' to 3'. And after having built that molecule of complementary DNA, another molecule that is complementary to the complementary DNA is formed, so that at the end of the day, we would have a double-stranded DNA molecule that will be inserted into the genome of the host. So this enzyme, reverse transcriptase enzyme, is a very important enzyme for retroviruses. Similarly, there is another importance for reverse transcriptase enzyme during cloning of human genes or during obtaining several copies of any human gene. For example, if you take you have insulin, a sequence that represents the insulin gene, and you want to get several copies of that sequence, we call this cloning. And we sometimes clone human genes inside bacteria. As the bacteria replicate their own DNA, they replicate the plasmid that carries the gene in sequence, for example, the gene for insulin. But the problem is that when the bacteria want to translate a messenger RNA of human source, or if they want to replicate DNA of human source, the introns represent a major problem for the bacteria. They do not understand what introns are because there are no introns in the genome of bacteria. The genome of the bacteria is already very small, and it cannot afford the presence of introns, and that's why those introns represent a major problem for bacteria, and that's why when we want to clone genes, we had better clone genes that have no introns. And the best way to do this is to use a cell where the gene in question is active and produces messenger RNA. So here, if I, for example, want to clone a gene for insulin, then I'll take a pancreatic cell, and that pancreatic cell actively produces insulin, and therefore the gene for insulin in that pancreatic cell is expressed, is on, and it forms the complementary sequence of messenger RNA in the nucleus. And that complementary sequence of messenger RNA will go to the cytosol, but after having been processed and after having been converted into mature messenger RNA that does not have introns. And then we will take that mature messenger RNA or that fully processed messenger RNA, and we will introduce that fully processed messenger RNA into the bacterium. And since the code, the genetic code is universal, then and since messenger RNA contains only introns, therefore all the codons on messenger RNA will be translated inside the bacterium into amino acids, and the bacteria will act as a factory for producing the polypeptide chains of insulin. Okay. And now we will be talking about the regulation of gene expressions in eukaryotes. We know that in prokaryotes, if we are talking about the prokaryotes, then the regulation of gene expression occurs at the level of the operons, or occurs through operons. And through these operons, we can regulate whether the gene will be induced or will be repressed, whether the genes will be on or off. But in eukaryotes, we don't have operons in eukaryotes, and the regulation of gene expression or the regulation of translation of a gene into a polypeptide occurs or can occur at several levels. So it can occur, of course, we're going to go over the diagram rapidly, and then we're going to read the corresponding slides. It can occur at the level of chromatin. For example, you all know that methylation of DNA prevents replication of DNA, and what matters here concerning regulation of gene expression is that it prevents transcription of DNA into messenger RNA. So this diagram, actually, I should have added the title. This diagram represents the regulation of gene expression. Regulation of gene expression, meaning that it represents the regulation of all the steps starting from transcription of the gene into messenger RNA to translation of that messenger RNA into a polypeptide. So you can control gene expression while you are still at the level of chromatin via methylation or demethylation of DNA. You all know that methylation of DNA results in silencing of DNA, and this is one way imprinting occurred in heredity, and that demethylation of DNA activates DNA and helps unwinding of DNA, and therefore exposure of the bases of DNA for the process of transcription. Similarly, acetylation of histones will activate the process of transcription of DNA. So at this level of chromatin, we can regulate gene expression through histones and through DNA. Both histone acetylation and DNA demethylation will help unwinding and loosening of the nucleosomes and of the folds of DNA around histones, and will result in easy transcription of DNA. We can also, of course, control gene expression at the level of transcription by affecting the transcription factors that activate or inhibit the process of transcription. After we get the primary transcript that contains both exons and introns, we can regulate gene expression at the level of RNA processing. RNA processing means that we're going to add a 5' cap and a polyadenine tail at the 3' end in order to protect messenger RNA against cytosolic enzymes, but it also involves the removal of introns. You can see here that we removed all the pink sequences, which are the introns, and now we have a shorter messenger RNA, which consists only of the exons that will be translated into amino acids. At this level of RNA processing, we also have a very important process that is called alternative splicing. And alternative splicing is one important method of changing the way we remove introns from messenger RNA, resulting in obtaining different isoforms or lengths of the messenger RNA, as we will be seeing on the next slides. And then it is transported to the cytoplasm. Of course, we can also regulate at this level. And then after messenger RNA has been translated, it becomes degraded. So when exactly will messenger RNA be degraded? Will it be degraded just after having been translated, or a bit later, or will it never be degraded, as in red blood corpuscles, for example, through this level of degradation of messenger RNA? The cell can control the lifespan of the messenger RNA, and therefore how many times a messenger RNA will be translated. And then finally, after the polypeptide has been translated, or after messenger RNA actually has been translated into a polypeptide, there is cleavage. For example, we can remove methionine. As you know, methionine is the first amino acid in all polypeptide chains, but if the cell or if that particular protein does not need methionine, then we will excise it. We will cleave the polypeptide. Sometimes cleavage is not only for removal of methionine, but cleavage is for removal of a part of the molecule that hides, for example, the active site on an enzyme, and therefore might result in activation of an enzyme. There is also chemical modification by adding certain groups or removal of certain chemical groups, and this is one of the famous functions of Golgi apparatus before packing. And then we have transport to cellular destination, whether intracellular or export to the outside of the cell, which is also a function of the endoplasmic reticulum and Golgi apparatus. Here we also have the protein has to be folded if we want that protein to acquire a three-dimensional shape, and therefore chaperone proteins will play an important role here in regulation of the folding of the proteins. And at the end of the day, when the protein has accomplished its function, it will be degraded. At any of these, or at each of these levels of gene expression, the cell can control the process by allowing it or by inhibiting it. So as we said, the regulation of gene expression in prokaryotes occurs through operons, but in eukaryotes, which are more complex, the process of regulation, of course, is much more complex, and the complexity here comes from the very numerous levels of gene expression, and at every level we can have a tool or a way to regulate that level or that step. And the importance of all of this is that it explains why, although all somatic cells, as we have said plenty of times before, all somatic cells in our body have the same diploid number of chromosomes, they have the same diploid genomes, yet some cells can carry out certain functions, others cannot, although they have the same gene. And we always say the famous example of the pancreatic cells, for example, that where the gene for insulin production is on, whereas the gene for melanin production is off. And the opposite occurs in skin cells. Although they have the same genome, yet the gene for insulin is off and the gene for melanin pigment production is on. Why does this occur specifically in certain cells? Because this gene expression, or the fact that the gene is turned on or off, is regulated by environmental factors, whether they are external environmental factors or internal environmental factors and signals. And when we say that the gene is on, this means that the gene is active, that the gene is expressed, and in order for the gene to be expressed, it has to be first transcribed and then translated. So as we have said before in the diagram, the regulation can occur at the chromatin level. We know that one of the major characteristics of eukaryotic DNA, that is that the eukaryotic DNA is wound around histone proteins. Every 8 histone proteins plus one stretch of DNA or one segment of DNA represent a nucleosome. Of course, you know that the histones carry a positive charge on some of their amino acid residues, and this what allows firm or strong attachment between the histones and the negatively charged phosphate groups. So if the histones carry a positive charge and the phosphate groups carry a negative charge, then there would be an electrostatic attraction force that helps stabilization of the turns of the DNA molecule on the histones. But if we make any change in the histone structure, then this would affect the whole configuration of that chromatin. You know that chromatin means DNA plus nuclear proteins, okay? For example, we can make the DNA more tight, the DNA turns tighter, and if they are more tight, then this means that this will inhibit the process of replication, the process of transcription, and any process that requires that the two strands are separated and the bases are exposed. On the other hand, if you loosen the attachment between the histones and the DNA, then the coils would become less tight. There is opportunity for the nitrogenous bases to be exposed and for the enzymes of replication or the enzymes of transcription to be able to read the template DNA strand. One of the major ways we can regulate at the level of chromatin, we can regulate gene expression is acetylation of the histone tails or addition of the acetyl group. So here, this is actually CH3, C double bond O, and this is an acetyl group. When you add an acetyl group to histones, you loosen the chromatin and therefore you permit transcription. So if you start by acetylation, and of course I mean acetylation of histones, then you will have a loosening of the chromatin or looser chromatin. And looser chromatin allows exposure of the nitrogenous bases, and the exposure of nitrogenous bases allows for transcription and therefore to continue the process of gene expression. But on the other hand, if you deacetylate, you remove the acetyl group, this will make the turns more tight and therefore will block the process of transcription. Here in the upper part, we have the same gene, but in the upper part of the diagram, we have the gene switched on just because we have open chromatin. Open chromatin means extended chromatin, and open chromatin is active. And this is because we did two maneuvers, the cell did two maneuvers in order to untighten the chromatin or loosen the chromatin. Number one, we removed the methyl groups from cytosines, from DNA. So this is unmethylation or demethylation of DNA, demethylation of DNA. And at the same time, there is acetylation of histones. So here, all these white dots are dots where methyl groups were supposed to be present. So we removed all the methyl groups, and this helped loosening of chromatin. And at the same time, we added the acetyl groups. We acetylated the histones. We added the acetyl groups. Okay, And these, of course, are transcription factors that would help the process of transcription. On the other hand, if DNA, if cytosine nucleotides in particular become methylated, you see the red dots are the methyl groups. And if we have removed all the acetyl radicals, then the gene is switched off because DNA is now deacetylated and methylated. So we always talk about acetyl groups for histones, and we always talk about methyl groups for cytosine bases on DNA. Methylation of DNA inactivates, but acetylation of histones activates. So they are opposite to each other. So again, addition of a methyl group to DNA silences DNA, and that silencing might be temporary or for long periods of time. On the other hand, if we remove the methyl group, if we demethylate, then the gene becomes turned on. And on the slide, you have two examples of long-term or permanent inactivation of a particular gene. For example, inactivation of one of the two X chromosomes in the somatic cell of a female. And in such a case, we obtain the bar bodies, which are condensed X chromosomes that are condensed smaller in size and that are eccentric in the nucleus. This is permanent inactivation of the X chromosome. And similarly, the genes that are necessary for embryonic development and differentiation of undifferentiated stem cells in the embryo. These genes work only during embryonic development, and then after birth, postnatally, they become permanently silenced, because afterwards what is really needed is growth of the tissues rather than differentiation of the tissues. Here again, we can see how cytosine bases become methylated. So we usually add the methyl group to the cytosine base, you see here, to the cytosine base. And these are the methyl groups, the brown things here, the brown structures. And methylation of DNA silences DNA. So this is silenced DNA. When we remove the methyl groups, then we have unmethylated DNA. So it is loosened, it can be transcribed, it is no more silent. Similarly, we added acetyl groups to the histone residues to help loosening and transcription of the DNA stretch. Now, methylation and acetylation, the processes we have just talked about, are examples of epigenetic regulation, meaning that this is regulation of gene expression through environmental factors. Here, methylation and acetylation are inner environmental changes, but there might be other changes that are external environmental changes like stress, like diet, but even it is related to the diet of the mother during pregnancy, which we call prenatal nutrition. So if your diet after birth, after you are born, affects you, this is what we call postnatal nutrition, how the newborn is nourished after a birth. This is what we call postnatal nutrition, but how the fetus receives nutrition, or what kind of nutrition does it receive before birth, this is what we call prenatal nutrition. All these examples, methylation, acetylation, stress, diet, free radicals, prenatal nutrition, all these are examples of epigenetic regulation. In epigenetic regulation, you affect gene expression, but you affect gene expression through environmental factors. You affect the phenotype or gene expression through environmental factors. This is what we call plasticity. So epigenetic regulation is related to plasticity, because plasticity is a change in gene expression due to environmental factors. and at the same time, you didn't make any change in the sequence of nucleotides on DNA, and that's why epigenetic regulation is completely different from mutations. It's not a type of mutation. We can also have regulation at the following level, which is the level of transcription itself. So the main enzyme for transcription is the RNA polymerase that is gonna transcribe a certain stretch of DNA, but it should first bind to a promoter and a particular sequence on the promoter that is called the Tata box. In order for the DNA polymerase to bind with the Tata box on the promoter, there are factors, transcription factors, like those one that are considered enhancers because they stimulate the binding of or facilitate, together with, of course, that complex of proteins that is called pre-initiation complex because it helps in the initiation of the process of transcription. This pre-initiation complex, together with the transcription factors that are enhancers, should be present here in order to facilitate the process of binding of the RNA polymerase. Of course, if they are enhancers, then their absence would result in inhibition of the process of transcription. We also have regulation after transcription, which we call regulation at the post-transcription level, and I'll be explaining it better on the diagram. But the main idea of the regulation after transcription, or the main concept we are going to explain, is alternative RNA splicing, meaning that if you have immature RNA, you know that the immature RNA has exons and introns, and that the introns have to be removed in order to make the messenger RNA more efficient, in order to make the messenger RNA all translatable, in order to make the messenger RNA shorter. But we are expecting, when we remove all the introns, to get the same kind of mature messenger RNA. But in alternative splicing, we get different types of mature messenger RNA, but that are closely related. For example, if you look at that diagram, we have a gene, and that gene will be transcribed into, it's a structural gene, and it will be transcribed into messenger RNA, which we call the precursor messenger RNA, or we can simply call it immature RNA. Why is it called precursor? Because it's gonna give rise to the mature messenger RNA. Of course, if we think straightforward, then we would think that we would remove all the introns, and that are, because they are non-coding, and keep all the exons because they are coding. So we are expecting the mature messenger RNA to be made of 5 exons, 1, 2, 3, 4, 5. But what happens actually is that every mature messenger RNA in such a case consists of only 4 exons. Not only this, but one of them consists of the exons 1, 2, 4, and 5, meaning that exon 3 does not figure on the mature messenger RNA, and which means that the enzymes have considered this exon and intron and have removed it. Whereas in the other form of the mature messenger RNA, we have again 4 exons, but these exons are exons 1, 2, 3, 5, meaning that this time it is exon 4 that has been considered an intron, and it has been removed as if it were an intron. So at the end of the day, even after adding the cap at the 5' end and the polyadenine tail at the 3' end, we have 2 very close to each other messenger RNA isoforms, isoform 1 and isoform 2, and they are translated into 2 different proteins, but those proteins are that are labeled here isoform 1 and isoform 2. It's true that they are not exactly similar to each other because they result from translation of different exons. However, like 3/4 of the exons are common. So they have in common 1, 2, and 5. And as we said before, sometimes there are certain changes in the amino acid sequence of the produced polypeptide that does not result in a major change in the function of the produced polypeptide. So actually we get polypeptide that we call protein isoform 1 having function A, and the other one, 2, having function B, but we have to take into consideration that function A and function B are very close to each other. Now, the 6th level of regulation of messenger RNA is, of the process of gene expression, is the regulation of degradation of messenger RNA. So, the span of time after which gene transcription, the span of time after gene transcription, meaning what gene transcription? Gene transcription means that we use DNA to form messenger RNA. So this is an important event in the cell. And there is another important event in the cell that is messenger RNA degradation. So one of the ways you can control the process of gene expression is via controlling this span of time between gene transcription and gene degradation, because the longer the messenger RNA molecule will stay in the cell, the more polypeptides will be produced by that messenger RNA. Okay, in human beings, a messenger RNA molecule might stay, for example, being translated in the cell for hours or even sometimes for days before being degraded. In certain structures in the body, like the red blood corpuscle, for example, the messenger RNA stays forever until the lifespan of the red blood corpuscle ends after 4 months, for example. Why? Because the red blood corpuscle does not have a nucleus, so it cannot transcribe a new messenger RNA if that messenger RNA becomes destroyed. And you might be asking yourself, then if it doesn't have a nucleus and it doesn't have DNA, then where did messenger RNA come from? Actually, this non-nucleated red blood corpuscle is the mature one, and it arises from an immature red blood corpuscle that was nucleated and that contained DNA. Okay, in bacteria it's totally different, because in bacteria the process of transcription is rapid, the process of translation is rapid, the bacteria reproduce rapidly by binary fission asexually, and therefore messenger RNA will be destroyed within minutes after being synthesized. I'll keep number 7 to the last minute of the PowerPoint presentation, and I'll be discussing first the modification of the protein itself. So meaning that we have translated the messenger RNA molecule into a polypeptide, and now we want to modify the polypeptide so that it becomes actively functioning. For example, in cases of enzymes like pepsinogen enzyme, which is a pro-enzyme, and trypsinogen enzyme, both of them are inactive enzyme. And for example, in pepsinogen, if you expose pepsinogen to low pH, then you remove a sequence of amino acids from pepsinogen and you convert it into pepsin. This this blue sequence of amino acids has to be removed. It is called a masking sequence because it was masking the active site. And now pepsin is an apoenzyme, and it needs a coenzyme or a cofactor to become a holoenzyme. The same applies to trypsinogen, with only one difference, that trypsinogen is activated by being exposed to an alkaline medium. Similarly, insulin. Insulin is a hormone. Insulin is not an enzyme. So after insulin is released from the ribosome, insulin consists of 2 peptide chains. Then there is cleavage of the peptide chains in order to change the number of amino acids in them, and to convert insulin hormone into active insulin hormone. The last point we will be discussing is the non-coding RNA, and I'll pause for a minute and then discuss this with you. Hello again. The last level of transcription is, or the last factors that result in regulation of gene expression are the non-coding RNAs. And the non-coding RNAs, upon discovery of the non-coding RNAs, scientists have discovered a new role for RNA. And that new role for RNA is to control gene expression. That's why we say that this is the 7th level of control of gene expression, which is the non-coding RNA. Now, there are recent data that have proved that 90% of our non-coding DNA is actually transcribed. Although it is non-coding, yet it is transcribed into messenger RNA. And this is recent information. And the transcribed RNA, or RNA that is transcribed from non-coding DNA, will be called non-coding RNA. So if you have on your genome certain sequences that are considered introns, that are considered non-coding, this does not mean that these sequences will not be transcribed. This does not mean that those introns will not produce sequences of RNA, but that RNA will not be called messenger RNA. That RNA will be called non-coding RNA. Non-coding RNA. Okay? So, what's the importance of non-coding RNA? And of course, we said that the overall importance of the non-coding RNAs is that they control gene expression, and this was really revolutionary in this area of study of RNAs, because we knew the function of messenger RNA, we knew the function of transfer RNA and that of ribosomal RNA to a certain extent, and now we have a 4th type of RNA. So it is important to know that we have a 4th type of RNA, that is the non-coding RNA. Now, this non-coding RNA has certain characteristics and subtypes. The first characteristic is that these non-coding RNAs can bind to specific types of proteins called Argonaute proteins. What are the Argonaute proteins? They are any group of proteins that will help the non-coding RNA that binds to them to silence a gene. So actually, Argonaute proteins are responsible for gene silencing, but they are not themselves the main silencers of the gene, but they help in gene silencing by RNA interference, meaning that the non-coding RNA, binds to some of the Argonaute proteins and results in gene silencing. Gene silencing simply means preventing gene expression, and we call this RNA interference. So RNA interference is the process when certain molecules of non-coding RNA associated with certain Argonaute proteins result in silencing of the gene. And from all these pieces of information, we can deduce that non-coding DNA or introns are no more considered junk DNA as they were considered in the past, but now they are related to very important functions, not only the fingerprinting that we have studied in detail, but also silencing of certain genes and preventing their gene expressions through the non-coding RNA. So please retain this information that there is a broad group of RNAs that are called the non-coding RNAs. Now, the types of RNA that are non-coding are, number one, the microRNAs, but you have to take into consideration that all the non-coding RNAs are related to silencing of the genes or blocking gene expression by a process called RNA interference. The first group of non-coding RNAs are very small, very short. That's why they're called microRNAs and denoted miRNAs or miRNAs, miRNAs. They are single-stranded RNAs and they are about 22 nucleotides long, so they are not that long. They form complex with Argonaute proteins. What's their target? How do they inhibit gene expression? We said that all non-coding RNAs inhibit gene expression. So how do they inhibit gene expression? They form complex with Argonaute proteins, and their target is certain specific messenger RNA molecules. So the microRNAs, the extremely short microRNAs, just like any non-coding RNA, will block the process of gene expression. How? The microRNAs inhibit binding of the specific messenger RNA that they target, because every microRNA has a specific target molecule of messenger RNA. It prevents its binding to the ribosome. And it goes without saying that if the messenger RNA molecule does not attach itself to the ribosome, then it will not be translated. The second type of non-coding RNAs are the cRNAs or siRNAs, that are the small interfering RNAs. They have the same target. They want to block gene expression. They are small, like microRNAs, but they're a bit bigger than the microRNAs. And they also, as I said, block gene expression, but not by preventing binding of the messenger RNA molecule to the ribosome, but by cleaving the messenger RNA itself. Of course, if the messenger RNA is cleaved, then it wouldn't be able to attach itself to the ribosome. And even if it does, it will be translated into a very different protein. Now, both microRNAs and small interfering RNAs require molecules that are called the dicers. And the dicers are nothing but a nuclease enzyme. The nuclease enzyme that is needed by the microRNAs and the small interfering RNAs, I think this makes sense because their main function, especially the small interfering RNAs, is to cleave the RNA. Therefore, the dicers, being nuclease enzymes, they will cleave any double-stranded part of the RNA molecule. For example, if the molecule of messenger RNA is folded, or if in the folds of transfer RNA, they will break down the double-stranded RNA. So we will have a single-stranded DNA molecule. And one of these single-stranded DNA molecules will be degraded, and the other one will form part of what we call RISC. What is RISC? It stands for RNA-induced silencing complex, meaning that after having like cleaved the double-stranded RNA into single strands, then one of the strands will be degraded and the other strand will form the RISC. And the RISC is RNA-induced silencing complex that will help the small interfering RNA to break down messenger RNA. Which messenger RNA will be broken down? The one that is whose sequence is complementary to the small interfering RNA. Of course, the process is very complicated. You just have to know a very superficial data about small interfering RNA. But if you're interested more, I can send you, and I'll send you videos and diagrams about this part. The last type of non-coding RNAs are the PIWI associated RNAs, or the P or pi RNA. It is a large class of non-coding RNAs, and their main function is that they guide specific types of proteins called the PIWI proteins to complementary strands of RNAs that are derived from transposable elements. You know, the transposable elements are DNA sequences, and if those transposable elements dare to make RNA, then PIWI associated RNA will direct those PIWI proteins to that complementary RNA that is complementary to the transposon and will prevent translation of that RNA, and therefore this will protect the coming cells or the descendant cells from being affected by abnormal polypeptide chains that might be due that might result from translation of abnormal RNA coming from like transcription of a transposon. Okay, I'll be sending a video to show the non-coding RNA types and functions, and by this we would have ended the third part of the molecular basis of inheritance, and therefore the whole unit of the molecular basis of inheritance. I hope this video was useful, and you will be receiving an assignment related to this. Okay, thank you.